Hello everyone, welcome again. So today we are going to continue looking at Mendelian genetics, and we are going to look at examine how we can look for at mono and dihybrid crosses and examine if they follow Mendelian inheritance pattern. And we are going to examine how we can statistically analyze that using ChiSquare analysis. So let's go ahead and get started. So this is going to cover mainly chapter 6 from the Open Genetics book, and then it's going to have, I've added a link for ChiSquare analysis that I thought was especially useful that you can also use as well. This link can also be seen in the description box below. So just to kind of review some terminology and what we should be mindful of when we are looking at Mendelian genetics, we want to remember what alleles are. Alleles are essentially alternate forms of genes, so different flavors, right? Brown versus blue eyes, short versus tall, widow peak, no widow peak, all those things. Those are different traits, characteristics that are coded for alternate forms of the same gene at many times. So it's the same gene sequence, just a slightly different variation that leads to a different phenotype, different physical features that we see. They are always going to be found, if it's an allele, it's going to be found on corresponding loci, so same place on homologous chromosomes, that exact same junction. Now, how many alleles can we have? Well, typical diploid organisms like ourselves would only be able to take 2 alleles at a time, one from your mom and one from your dad. Those two can be exactly the same, or they can be slightly different variation, or they can be extremely different variation. But either way, you can only have 2 alleles present in any of your given cells based on the genetics that you have. The half of them are coming from obviously your egg, mom's egg, and the other are coming from the sperm that when combined together at fertilization will give you your fully fertilized egg in the form of its zygote. So these alleles, like I said, can be exactly the same, in which case they would be termed homozygous. So both the alleles at a particular gene locus from your mom and your dad have the same exact code, same information, so they code for the same thing. Both code for blue eyes, both code for short height, whatever the trait may be that you're looking at. On the other hand, in a heterozygous case, you are going to have those 2 alleles be different. So one will be for blue, one would be for brown eyes, one would be for short, one would be for tall. And so whatever that trait is that they are coding for, they're going to have different variation in a heterozygous case. Now, those many times we have talked about how they can be inherited, once they're inherited, how they can show. Sometimes you have dominant versus recessive, where one shows up predominantly and the other one is masked by it. Other times there is codominance, where both can be present and both can show in the final outcome. So the law of dominance tells that each individual has 2 alleles for each trait, one from each parent, and these 2 alleles, when they're different, one of the pair of alleles coding for that trait would be expressed, whereas the other is unexpressed. It's not that the other is unexpressed, but the other is masked by the dominant phenotype. Now, we already know that's not always the case, right? But it is a good starting point to work with. Now, as we're inheriting these traits, the law of segregation, which we've talked about before as well, is that each trait, each locus, each gene is going to have the 2 alleles separate, independent of anything else that's going on in that chromosome, in that genome. So during the formation of your gametes, eggs and sperms, the 2 alleles will separate from each other from one another, and they may recombine at fertilization to produce the genotype for the final trait that you see in the offspring. And this happens completely independent in each case. Now, that doesn't always stay true either. There are always exceptions to the rule, and there is a reason why there's an exception. We'll learn about those during linkage when we talk about how presence of genes that are close to each other can affect their ability to get moved from one cell to the next, to the progeny. So, but today we are going to look at illustrating genetic crosses with Punnett squares, and then look at what type of genotypic and phenotypic frequencies we would be expected to see, and what we observe, and once we observe something different, or in our experiment, how do we account for it statistically? So for genetic crosses, we talked about before last lecture how Punnett squares is one way that we can simply demonstrate how the different alleles are getting transferred to the progeny in a diagram form, in a graphical way. So it is a simple diagram that helps us see the different combinations that in genotypic terms, different genotypes that can occur in the progeny from a particular father and mother with a particular genetic background. It also helps us visually see the odds, the percentage of the offspring that will have one trait versus the other. And so in this case, let's look at this. So you have your father's genes and your mother's genes at a particular locus. In this case, the mom is homozygous recessive because it's lowercase b. And then your father, in this case, is heterozygous. So it has a dominant and the recessive allele, which means that the dominant phenotype is what you would see visibly outside. When you look at the progenies that are possible, you will notice that 50% of the progeny is going to look like dad. So one dominant and one recessive allele, and 50% will look like mom, being completely recessive. Now, when we look at the gametes that these individuals form, so in looking at one of these chromosomes right here, you have a chromosome that is heterozygous at the particular locus that we are looking at. So that's depicted in this way. And when in meiosis you're forming those haploids, egg and sperm, each one of those will duplicate and then get in that first reductive division, you're going to have one of those homologous chromosomes go to one egg or one cell, and the other to the second, and then each one of those will further separate in the next round to form the individual gametes. So each one of these have one copy of the allele. Half of them are going to contain one of the alleles, maybe it's the dominant one, and the other half will contain the recessive one. Okay, so let's look at a monohybrid cross. It's going to deal with a single gene, very similar to what we've looked at last time. So the basic idea in monohybrid cross is that it deals with one single gene. And that one single gene is going to be presented by two letters, both of which are the same. If it is homozygous dominant, it's going to be represented by two capital letters. When it is homozygous recessive, it's going to be depicted by lowercase a's. And when it's heterozygous, it will be a capital and a lowercase a, not AB. So you're not taking different alleles, giving different alleles different letters. It's the same letter, just capital or lowercase. So in this case, you need those two alleles, obviously, to make that one gene. So here is a monohybrid cross. So remember, in meiosis, you're going to have that genetic information. So half of the gametes formed from the dad is going to have the dominant A allele, and then half of them will have the recessive lowercase a allele. And so here you have your mom and dad. They are both heterozygous for this particular gene locus, and so you create a monohybrid cross, Punnett square, and you see 1/4 of your progeny is from this cross would be expected to be homozygous recessive. The rest of them will show the dominant phenotype, but only one of them would be homozygous dominant, and the other two, 50% of the progeny is going to be just like the parents, so heterozygous for the dominant. So here again are the possible gametes from the dad and the mom. a capital A and the lowercase a. And once you follow through with it, you will see that the genotypic ratio is going to be different than the phenotypic ratio. So, once again, in the phenotype, 3 of them will show the normal phenotype. In this case, 1 of them is albino, which is the recessive, so it's 3-to-1 phenotypic ratio. So that's what the outward appearance is like, what the physical expression of these alleles looks like, that we can follow, that we can score. That's a dependent variable, so to speak. And genotypic ratio is going to be 1 to 2 to 1, since we have a quarter of the population as normal, a quarter as albino, and the remaining half as heterozygous for albino. So here is another example of a monohybrid cross. So we can kind of follow through with it and see if we can look at it statistically as well. In this case, we're going to use fruit flies, Drosophila melanogaster, and we are going to be using a special type of fly. So we're going to have a female that is pure breeding for Apterous wings. Those are basically all shriveled up tiny wings, so they can't really fly very well or at all. And the males are going to be pure breeding wild type wings. So they'll be completely normal the way they should be. In this case, we are representing this particular genotype a little bit different way, which I had also talked about previously in the last lecture. So the AP is both lowercase for Apterous, and the plus sign indicate the wild type phenotype. And AP, which is not the wild type, but actually the shriveled up wings, the Apterous, is just simply signified as AP. Many times genes are named because of a particular physical disability or phenotype that people see in the mutated form or mutant form rather than the wild type form. And then later on, when the wild type is found, you may see this type of notation where the plus indicates that it is the normal wild type function of that. So in this case, the first cross, the F1 generation cross, is going to be between the progeny from the parental cross. So when you have a pure breeding Apterous crossing with pure breeding wild type, all the progeny is going to be heterozygous for heterozygous wild type, showing physically wings. So both the F1s that are going to be crossed in the next generation will be heterozygous at the Apterous locus. So here you have F1 female, which is AP plus wild type and Apterous, and then male, which has again heterozygous the same way. Here is an example of what this would look like. So here are some wild type flies. This is a female, this is a male, and you can tell that this is a male because of this black at the behind. They also have sex pads usually on their legs, and then their wings are nice and full so that they would be able to completely fly the way you would expect them to. And here is Apterous male fly. You know, it's a male. They have a slimmer body. The females have a more plump white abdomen, and then it has those shriveled up wings on the top. So, okay, so let's say that we were doing a cross where we were using these wild type and Apterous flies, and we make that parental cross, and then in our F1 generation we cross that F1 generation and we place that cross, and then we examine the progeny. What we would look for in that case is that we would expect for a monohybrid cross that Mendel's rules would predict a 3-to-1 dominant to recessive phenotypic ratio. So we would examine if our numbers fit these expectations. How would we do that? Well, of course, that would require some statistics. So let's look at what type of statistics we can perform in order to do that. Typically, what we examine in genetics, this type of Mendelian ratios and Mendelian crosses, we examine its how close we get to expected ratio using ChiSquare Test or goodness of fit. So it's a statistical test. It is used to compare observed versus expected or hypothesized distribution. And then we can look at how close we got and if we were in that window, which would be expected. In our case, our expected distribution would be derived from Mendel's ratio, so those 3-to-1 expectation. And we are going to do that by observing distribution based on a whole class's fly count or whole experiment's fly count. So multiple crosses, not just a single one. So anytime we are using statistics, we have to have our hypotheses derived first. There are, so we have our main hypothesis that is going to be dependent on what is expected based on Mendelian ratios. And then we're going to have our null hypothesis. The null hypothesis is essentially going to be that the observed distribution is not different from the hypothesized. So basically it's that our hypothesis is correct. Our hypothesis is going to follow Mendel's ratio, and it is going to look just like that. It's going to match those expectations. Our alternative hypothesis, HA, is basically that our observed distribution do not follow Mendelian ratio, do not match those expectations, and are somehow going to be different than those. And so in that case, you would need to then think of reasons why that was the case, whether it was that one of the phenotypes or one of the genotypes was lethal or something else was wrong with it, or we missed it. What could be the reason that if maybe we didn't make the cross correctly, that our hypothesis did not come true. So we would want to then think about both technical as well as biological reasons why it didn't work out. So when we are running ChiSquare test, there's several assumptions that we are making. Our first assumption is that every gene locus that we are observing in a particular experiment is independent of other things, are independent of each other. So any phenotype that we observe in one fly is not going to affect any other fly around them, and it's going to be individual to each fly. They're all independent. It's not going to have any cross interference. The second is that ChiSquare test use categorical data, right? So that basically means that we need to have actual data for it, actual numbers for it. We can't just be using with clear-cut categories, right? So we have to have a very clean categorical markers, clear dependent variables or clear phenotypes we're observing. So it has to be, male versus female, blue versus brown, anything like that that is differentiated, not differentiated, something very clear that we can observe and categorize as well as follow. And then finally, the number of samples observed in each category are expected to be at least 5. So you need to have a significant N number. You can't just make one observation and be done with it. You have to be able to do multiple replicates and multiple observations, which is why you would want to have more something that can produce more progeny and multiple crosses going at the same time, so you can have more data collected. The chi-square itself, the formula is essentially the sum of all the observed phenotype for each one of the categories that you were observing. It would be the observed minus the expected numbers of that particular category squared divided by the expected numbers, and then we add up all of those calculations that we get. So we sum them all up, and that number that we get is the chi-square value. Once we have that number, there is a chart that we examine to look at where it falls and whether it is going to be in the significant category or not, so whether it will follow the Mendelian genetics or not. So let's say that when we observe the flies, we get 325 wild type flies, and we get 107 apterous flies. Now, that would the first thing we would need to know is how many total flies that we are observing to get these numbers. So we just simply add those two up. In this case, it is 325 and 107, right? So you're gonna get 432 total flies, right? So I'm just gonna write T for total is equal to 432. I could have made it a lot easier, but I don't know. Now, if we were observing 432 total flies, we would expect Mendelian genetics would expect us to get a 3-to-1 ratio, which means that we would have expected in real life for matching Mendelian ratios, we would have expected to see 108, right? Apterous things, flies, which are the recessive, and we would have expected to see 324. So we can already see this number is very, very, very close to what we observed, 324 of the wild type allele. Now let's say instead if we were to see, in another experiment, another person observes the flies, and in their experiment they find 275 wild and 25 of the apterous. So they find a total of 300 flies, out of which 275 are wild type and only 25 are apterous, right? You would still want to do the same thing. You would want to see how many you would expect to see. So in this case, when you do the calculation, you would have expected to see 75 flies that were apterous, not 25, and you would have expected to see 125 or 225 rather, sorry, wild type flies, right? So this just tells you how you're going with that, right? Okay, so I'm gonna stop this somehow. Okay. Ah. Okay. So going to the next one, and let's see what happens. So, I'm trying to find my cursor again. It refuses to come back. It's that. Okay. Oh well. So let's look at that. So our observed again, since our numbers have gone away. Okay, so like we said, let's look at our numbers again for a second. So we said that we had 275 observed and 25 apterous in one of the scenarios. and in the other one we had 108 and 300 and something. 1 second. I have those numbers. 108, 432, 324, right? So you can get your total expected based off the Mendelian ratio, 3/4 wild type and 1/4 apturus. And you can get those by simply then taking your full number and multiplying by 0.75 or 0.25, or, to get those actual numbers. And what we had found was, I wish I can go back so it can show me. Okay, so once we get those numbers, it's not moving. My computer has melted. Sorry about that. Oh, there was some weird thing that was going on. Okay, so 325 in one case and 275 in the other, and then 107 and 25, and our expected in these two cases would have been 324 and 225 and 108 and 75. Okay, so let's move on. Okay, so now we want to know, well, visually looking at it, we can already see where the issue is, but now we can examine it a little bit closer and see if our data matches expectation. So what we would need to do is follow through with that formula of observed minus expected squared divided by expected, and you add up for all the phenotypes. In this case, it's pretty simple because it is all we have is one, these two phenotypes are two categories, wild type and apturus. So in this case, if we were to work it out, right? So it's going to be, for example, for one of the first category, right? And I'll show you an example, observed, which is 325 minus expected 324. And then we're going to square that. So the answer is 1 square divided by 324. Right? And so you would get that answer. You would do the same thing for the second one. So, it should be 107. So it's 1 square over 324. So it's 1 over 324 basically. And then plus the second one, which is 107 and 108. Again, that's a difference of 1 square over, yes, plus the total from the second one, which is going to be again 1 divided by 108. And the answer in this case is equals 0.012. So in this first case, the chi-square value comes out to be, chi-square is equal to 0.012. 0.012. 1, 2. Let's look at the other one because the other one is a lot different, right? So it's going to be 275 minus 225. That gives me 50 square divided by 225. And I get 11.11. And then we are going to do the same thing for the others. So 25 minus 75. And then divide that by 108, by 75. And I get 33.33 plus 11.11. So I get 22.22 because one was a negative and one was a positive. And in the second case, I get a 22.22. So now we are ready to look at the chi-square chart, and we are ready to see if our numbers match the expected expectation for these values. So to determine that, we first need to figure out the critical value on the chi-square table. It has two different sides. So one of them is your probability, which is along the top, right? So this side is your alpha value. So that's your p-value essentially. 0.001 is going to be the lowest on this particular chart. 0.05 is essentially equivalent of the 5% FDR, 5% false discovery rate, which is the typical value that we examine when we are looking at statistical significance. So our value should be at or below this number in this column, whatever number is written in this column. And the second side of the chi-square table is the degree of freedom. The degree of freedom is essentially equal to n minus 1, where n is the number of categories that we have in our analysis. In this monohybrid cross, the n number is 2 because you have two categories, wild type and apterous. So the degree of freedom is going to equal to n minus 1, 2 minus 1, which is 1. So we would go from degree of freedom to 0.05, and our expected chi-square, if it matches Mendelian genetics ratios, expected ratios should be 3.84 or below. We had a 0.012 in the first case, so definitely that fits. So in this case, in this particular experimental observation, yes, it matched Mendelian genetics ratio, expected ratio. In the second case, it didn't. It was way higher than that 3.84. Right? So that just lets you know if you were in the right place with that particular gene locus, if there were no interfering genes, no other genes that could have also contributed to it, in a linked manner. So the critical value versus the calculated value. The critical value is basically your value that that we just highlighted. So if your calculated value is less than or equal to the critical value, then there is no statistical significant difference between your data and the hypothesized distribution, which is the one that you were expecting. If the value is equal or greater than the critical value, then there is a difference and your hypothesis was not correct. That means that there was no, that it did not match the Mendelian genetics. So you want it to be less than that. So it's pretty simple when it's just a simple monohybrid cross. But as soon as we get a little bit more complicated, where you're looking at multiple phenotypes and multiple gene loci at the same time, it gets a little bit more complicated. So we're going to start, we're going to look at a dihybrid cross, which essentially is two genes now. So two separate genes. Each gene is going to have its own letters to represent it. And according to Mendel's law of independent assortment, we say that the pair of alleles will segregate independently of each other during gamete formation. So they should not have any cross interaction with each other, or they shouldn't be dependent on them. So we should be able to look at two separate ones and still have them segregate separately and independently of each other, giving us, again, specific Mendelian ratios. So in a dihybrid cross, here is an example with a heterozygous individual for those two gene loci that we are interested in. So they have a dominant and a recessive allele for both. To look at the various gametes that can be formed, you do a FOIL method, and you look at all different possibilities of the various gametes that can form. So when we do that, the mom genotypes is AA, capital B, little b, sorry, there's a typo. So the possible gametes are going to be 4 different ones. You can have both dominant alleles, both recessive alleles, small recessive for the first and a capital for the second, and a recessive for the first, second, and dominant for the other. So capital A, capital B, capital A, lowercase b, lowercase a, uppercase b, and both lowercase. Now for the dihybrid cross, again, the Punnett square works the same way, but it is a lot more complex. So there are 16 different genotypes to examine. Some of them are going to be the same. So you put all 4 gametes on both sides, one for the mom, one for the dad. In this case, both mom and dad were heterozygous at both loci, so you have the same four gametes, and then you fill it up by going up and across exactly the same way as you would with a monohybrid cross. So you're going to place the dad gametes on the top, the mom's gametes along the side rows, just like we did before. And then we're going to cross the formations to get offsprings with 4 letters. Obviously, the gametes are haploid, so they are 2 letters. Offspring are diploid, so they are 4 letters. And then we examine what type of genotypes and phenotypes we are going to get as a result. The first thing we want to do is make a list of all the unique genotypes that we see in our cross. And here is that list of all those different genotypes that we observed. There are 9 different genotypes that you can get from a dihybrid cross, and they're all listed along this side. Following that, we then go ahead and count how many offspring would have each one of these specific genotypes and write those down. So there's 16 possibilities right here. For each one of those 16s, you see how many of these genotypes are represented, how many times out of the 16. So, okay, so once we have put the actual numbers associated with each one of these genotypes, these are those numbers. Next, you are going to be looking at what those mean, what exactly those genotypes are going to be representing at the end of the day. So in this case, we are looking at the various genotypes associated with attached and brown, attached and blue, unattached and brown, unattached and blue. And those were the various possibilities that we could have observed inside our specific dihybrid cross in this place. So right here, you can see the phenotype associated with each one of the genotypes. And so you can see that there are a lot of genotypes that give the same phenotype, as expected, because you have a dominant and a recessive allele for both. So we can calculate the total phenotypic frequency by adding the sums of your genotypic frequencies that lead to that particular phenotype. So all these that give attached and brown can be combined together, and you get 9/16. 9 out of the 16 offsprings having those particular phenotypic outer appearance. And then 3 out of 16 with 1 of the dominant and 1 heterozygous, 1 recessive phenotype, and another 3 out of the 16 with the other dominant and the first recessive, and then only 1 out of the 16 with a complete recessive phenotype. So in a dihybrid cross with 2 heterozygous individuals for those 2 loci, you would get a 9:3 to 3:1 ratio. That's what you would be expected to see. So you can try something like that on your own using that link in the description below. It is a great site that walks you through ChiSquare in great detail and with a lot more information for those who are interested in it. And it also provides you with example problems as well as places for you to calculate them. It has tables that can be changed and customized and to run your calculations and make sure that you understand the concept appropriately. So in this case, I'm giving you an example with a parental cross where it is a pure breeding purple and smooth corn that is crossed with a pure breeding yellow and shrunken. Both of those are recessive corn phenotype, corn plant. And then the F1 generation, which is heterozygous for both of those loci, is allowed to cross together. The gametes in that case are going to be the same as what we just talked about. Um, some of them will contain both dominant alleles, some of them will contain one dominant and one recessive, and then some of them are going to have both recessive alleles for the two separate genes. When we cross it out, once again the same way, you have the gametes of the dad, so-called, and the gametes of the mom on the other side. You can look at the expected number of phenotypes in any given case by looking at how many different phenotypes you're seeing here. And so it's 2 to the power of 2. So there are 4 possible phenotypes because there were 2 different genes and 2 alleles. So purple and smooth, purple and shrunken, yellow and smooth, and yellow and shrunken. And you see the examples of each up here. And the expected number of genotypes would be 3 to the power of 2, which would be 9. And so right here, you see that cross played out. Only one is recessive completely for both alleles, for both genes. And there's one that's completely dominant for both genes. And then several others that are either dominant or recessive for one or the other. So here you have been given a chart where you are putting in, we are provided with the observed numbers in the second column. And what you need to calculate is the observed ratio. You know the expected ratio, which is 9 to 3 to 3 to 1. And you know the expected total number, which is going to be 433, the total number of kernels that are counted, multiplied by the ratios expected for each one of those. So in this case, it's multiplied by 9 over 16, and you get those numbers. What I would like you guys to do is to, if you're watching this all the way, I want you to solve it out and write down in the comments below the chi-square value that you received and whether or not it followed the Mendelian expected results for a dihybrid cross. The chart for that you can see up above as well. So make sure that you take into account the degree of freedom and then the 0.05 probability. So I look forward to seeing your responses. The summary for today is that we went over how alleles of different loci are inherited independently of each other unless they are genetically linked, which we'll talk about in great detail in a couple of weeks. The expected phenotypic ratio of a dihybrid cross is 9 to 3 to 3 to 1, except in cases of linkage or gene interactions that can modify this ratio. And for a monohybrid cross would be 3 to 1. Chi-square is a statistical test that we use to examine the differences between these categorical variables from a random sample, and it helps us judge the goodness of fit between expected and observed ratios or results, especially when we are looking at Mendelian genetics. The next lecture that we are going to have is going to be all about mutations and how they happen and what is the result of these mutations before we delve further into linkage and pedigrees. So I hope this was useful. Talk to you soon.